unmute yourselves. And thank you for joining us.
All right, thanks, Sam. I think I've unmuted the microphone. I'm Dixie, and Mary Ann's.
I am Mary Ann.
Right. We realize that our names are flipped underneath of our photo, but we are actually coming to you live from Seattle, Washington, where we're in a hotel room sharing a little laptop computer. So hopefully this works out well, and you're all able to see and hear us. I'm going to go ahead and start with our presentation. Today we're talking to you about writing a great protocol, the opportunities and pitfalls that come up along the way. One of the quotes that I like to start with is from Bill Clark. He was the former director of the NINDS-CTMC at the University of Iowa. He's a longtime statistician who's been involved in a lot of neurological trials. And he gave a talk once, and it was titled, "Everyone Should Do a Clinical Trial Once." And that there are a lot of things that go into running clinical trials, and as you go through the process, I think you learn a lot on how to implement them and improve efficiencies and make sure that everything is done accurately. But part of that's based on the fact that each participant in a clinical trial, and there are many, it takes many players to pull this off. And everyone has a bit of a different perception of the reality of clinical trials. So if you start out, the marketing people think that the glass might be half full. The legal team think that the glass is half empty. The engineering people think that the glass needs to be re-engineered. There's something wrong with it. The lawyers have their perception. The PIs are PIs are often worried about what the other PIs are doing or what they're gaining from it. The subject wants to know if they're getting placebo or are they getting drug. The IRB has concerns about the glass being vulnerable. The FDA is concerned about the regulatory components of how the glass is being made. The coordinator is just worried about getting all of the work done. The media comes out with often some type of broadcast that may or may not reflect what is known to people who are participating. And the accounting people are looking at the numbers, and the public is generally sitting somewhere really not aware of all the operations that go into it. So what we're going to do is walk you through some of the logistical things that go along with operationalizing a clinical trial, and a lot of that comes down to having a good protocol written. We're going to talk to you so that you begin thinking about the logistical considerations that go along. And the goal is that there will be no surprises with the budget and no surprises with trial implementation. So I'm going to turn it over to Mary Ann now, who's 6 inches away from me.
Okay, I'm just going to go back one slide there. So thanks, Dixie. So I'm going to talk through, again, sort of when you're starting a protocol development, one of the things you need to do. The first step really is to develop a team. Team, as Dixie said, a clinical trial takes many members. So this is just a list of some of the team members that you're going to have to pull together when you're thinking about putting together a protocol. So you need a lead protocol coordinator or a project manager. That person, I often say, needs to be your best friend. They need to be somebody you communicate well with. They're going to help you organize all the other team members. They're going to help you throughout the entire process of the clinical trial. You need a sponsor. You need to know who's going to fund the study. You need to find out if you're going to be the sponsor from a regulatory standpoint. You need co-investigators. You want other investigators that have experience with this patient population to help you develop this protocol. You need patient representatives, and this is, I think, come to light more vividly in the last few years. But having some patient representation, whether it be from an advocacy group or a patient or family members, to really help and see whether a study is being developed and a protocol is being written that any patient would want to enroll in, or that they could meet the demands of in terms of how many visits there are, how often blood is drawn, those types of things. You need coordinating centers. As Sam introduced Dixie and I, I'm at the clinical coordinating center and Dixie's at a data coordinating center. You need to know how you're going to coordinate all of the activities in your study. You need vendors oftentimes. So if you're going to be doing any type of outcome measures, whether it be an MRI, or you're going to have a central reader, or an EKG, will you have a central reader, laboratory vendors, pharmacy vendors. So those are things you need to start thinking about. You want to have a steering committee. You want to have other, again, peers and other physicians thinking about how they can help you to steer your study to be successful. You also want to think about a data and safety monitoring board, whether you have a study that's funded through NINDS and it's going to use a DSMB that is convened by NINDS, or if you're going to convene one yourself. So those are all the team members you want to think about. And then step two. Step two is really to glue your team members together. So what will help to do that is, having a well-written protocol, having everybody on board with your well-written protocol, having good study materials and training and education, and thinking about that up front even before you begin to implement them. Having a well-designed data collection system. If you want to have a successful study, you want to collect the appropriate data to be able to reach an endpoint and make some conclusions from the analysis of your study. You need to have a well thought out data and safety monitoring plan. You need to have some standard operating procedures, some procedures that are going to guide how everyone performs things within your study. And of course, with our smiley face there, you need to have adequate funding for all of this, and creating a budget is a big part of this as well. You also need to really develop realistic timelines. Everything takes longer than you will ever imagine it will take. You will run into things that you would never have anticipated. And while we try to say that good coordination and having people that are experienced can help speed the study startup process or the execution process, there are always issues that come up. So making sure that you're planning for your study startup, planning for your how long it's going to take to execute your study. How long do you anticipate for it will take to identify all of the patients in the study? Then how long will each of those patients be in the study? And then how long do you need to do your data cleaning and make sure that the data is all accurate before you lock your database? And then you move into the reporting and analysis phase. And then, what you also need to do is have someone, such as your project manager or project coordinator, keep track of all those timelines to know, if you're slipping, if it's taking longer to recruit your patients, what is that going to do to your funding? How are you going to make it through the end of that study? So writing a protocol. This is really the first step. You're writing your grant, and oftentimes your grant application requires that you attach a protocol as an appendix. We have to really stress that the protocol is different than your research strategy. They have similar things in them, but your research protocol has very specific sections to it. There is a protocol template that's available on the NINDS website, and we have the link here. And what your protocol is, I really like this this figure on this slide, because really it is like having a recipe book. You want to think about your protocol as a cookbook. The better you describe how much flour, how much sugar, all of these pieces, the better off you're going to be able to operationalize that study. So one of the first things you need to think about is your study going to be regulated by the FDA? Do you need an IND, or is this a non-IND study? You can reach out to FDA, you can ask that question, you can engage them to determine whether or not you need an IND, you can look at the regulations. If you're using a drug that has already been marketed, it's already an approved drug, but you're just going to be using it for a different indication, then you can oftentimes request an IND exemption. So you want to think about that. If you are submitting an IND for your study, if it does need to be regulated, there is a guidance document that NINDS has out that requires that your IND be filed and submitted at least 31 days before your grant submission. And they have that 31 days for a reason. The FDA has 30 days to respond to an IND application to let you know if it's okay to proceed with the study or if they have major issues with the study. So the NINDS would want to know those issues before they would review the grant. So they want that to be included in your grant application, that you filed the IND and that you haven't received or have received feedback from FDA. So this slide just shows a list of all of the sections that need to go in your protocol. So if you look at that, you go onto the web link that was listed on the last slide for the protocol template that is on the NINDS website. These are all of the sections of a protocol. So what I'm going to do now is go through each one of these sections and just give you a little bit of insight about what you put in these sections and talk about each section. So you want to start the protocol off with your study objectives. You have your primary objective, which should really be to specifically address, your hypothesis. What is your hypothesis? That should be really your primary objective. You can also, you need to state the hypothesis in quantifiable terms. So, the experimental treatment will result in 12 months of additional survival compared to the control treatment. Those are the ways you want to state your objective. You can also have your secondary objectives, and these may or may not be hypothesis-driven. They may include some secondary outcomes, sort of exploratory things. But what you also want to think about with your study objectives is you don't want to try to fit everything in. Again, if we use the baking analogy, a cookbook analogy, if you try to put too many ingredients into a recipe, you end up with a disaster. So you really want to focus on what your primary outcomes are and, one or two secondary outcomes. If you try to fit too much into a protocol, the likelihood of success is lower. In the background section of a protocol, you want to provide the study rationale. What is the historical backgrounds? What patient population is being studied? Why is there a need for this study? What's the relevance? What is the supporting data? You need to list what your supporting data is. You can describe preclinical studies that have been done that have sort of led you to this current hypothesis. If there's any clinical results that also support your hypothesis, you want to put that in there, and you want to really justify the study. So to say why you're doing the study and why it makes sense to do the study at this point. Also in the study design section, you want to briefly describe sort of the design and how you're going to do the study. So what we put in here is just a diagram. Oftentimes it's good to use a diagram in the study protocol because it helps people to really visualize how the study will happen. So you have a screening phase. How long is that screening phase? Up to about 4 weeks in this example. Then you have a treatment phase and what happens during that treatment phase. There's going to be an active group. There'll be a placebo group potentially. How long will it go on for? And then the follow-up phase and how long the patients will be followed after they come off treatment. The next section is about selecting and enrolling subjects. So this is where you list your inclusion and exclusion criteria. You really want to focus on what is the actual patient population that you want to include in your study. This is a big area where you would want a patient or a patient advocate to be involved in helping to shape this portion of a protocol. Sometimes making a study too onerous so that patients wouldn't actually meet the entry criteria could lead to a failure of your study if you can't enroll the right subjects. But then if you broaden the enrollment criteria too large, then you may not reach the endpoint that you're looking for in a specific patient population. So you really want to hone in on what are the criteria that are going to define your patient population. You want to also list what are the criteria for subjects who withdraw from the study. Are they going to stay on study drug? Or if they withdraw from the study drug, do they stay on study? Are you still going to be following those patients? You want to list your enrollment procedures. How are you going to be recruiting patients? Are you going to have screening logs to identify the patients that come in and are screened for the study? What criteria make them ineligible for the study? It's often very good to capture that information so that if you're having trouble with recruiting your subjects, you understand why. If there's one entry criteria that might be too stringent, or you might want to think about opening up that entry criteria. You want to have a procedure for performing informed consent, so the informed consent process. You want to write out what your randomization or treatment assignments, how that is all going to happen logistically. Not just that it's going to happen, but what are the logistics of that you want to include in this section. So your study intervention. First you need to think about what is your intervention. So when we say intervention, we mean, is it a study drug? Is it a some medication? Is it a device? Is it something else? Are you doing a biomarker study? You want to describe that in this section about your study intervention. And then you need the details. Again, you want to think about this as a recipe. You need the details about how are you going to be handling your intervention. Who's supplying the drug? Who's supplying the matching placebo? How will that medication need to be stored? How should it be prepared? What will the labeling be like? How is it going to be dispensed? Do you need site pharmacists to dispense it, or can it be dispensed by a research coordinator? Are there any unmasking procedures? What happens if a patient needs to be have a drug dose change? Is that allowed in your study? Are there specific medications that a subject will need to be on in order to be eligible for your study? Those are required medications, or are there medications that they're not allowed to go on or not allowed to be on to be either entered into the study, or during the study, if they go on that medication, they would need to come off study. So thinking about, the medications that they're going to be on sort of in a concomitant basis. And then how are we going to assess compliance? How will you know that the person actually took the pills or, got the infusions? How is all that going to be assessed at the end of the study? Because if you're looking at your patients and you're looking at the active drug versus the placebo arm in the analysis, you want to think about how did they get all of the study drug? Did they have a dose reduction at some point? So you want to really write that into your protocol in advance and plan for it. I recognize you can't really read this slide, but what this is a schedule of activities. It's a very important piece of a protocol. What you need to list is a schedule of every single procedure that will be done on each subject, and that's really on the left-hand side of this chart. And then across the top, we list every single visit that the patient will come in for. And then there are X's in the boxes of, at that visit, that procedure will happen. So this is really what many research coordinators will use. People will use it sort of as their cheat sheet to know what is supposed to happen at each one of the visits. So it's very important to really think about this. This also helps to build your budget and thinking about how you're going to reimburse sites or get paid for different activities. So again, I recognize you can't really read this, but if you want to think about your schedule of activities, it's a very important part of your protocol. You want to have a section that talks about your clinical and your laboratory investigation evaluations. So you want to describe all of the procedures that are going to occur at each and every study visit. So what will be the procedures during the screening process? How will subjects be randomized? What will the processes be? And then for each, as I said, for each and every study visit, what will the procedures be, and how should they be conducted? If there's a specific way those evaluations should be done, if one test needs to be done before the next test for consistency, or because the patients will get tired after certain tests, you want to make sure that's listed in the protocol so it's done consistently, the same with all patients. You want to really talk about adverse events and how those will be managed throughout the study. You want to look back at your previous data with this drug. If there's preclinical information or previous clinical trial experience, you want to list everything that's expected in terms of adverse experiences. Does this drug have an effect on the liver? Does it have an effect on the stomach? Is it going to cause nausea? Does it cause dizziness? Those types of things are often listed either in a label of a drug that's already a marketed drug, or if there's a drug that's not yet marketed. If you're working with a company that has an investigator's brochure, they have data on common adverse events that have been linked to this study intervention. So you want to list all of those in the study, in the protocol. And you want to describe your procedures for study management and modification of the dosing regimen, the intervention regimen as well. If someone has an adverse event, are they able to half the dose, or do they need to come off the medication altogether? You want to list the criteria for study discontinuation. You need to define stopping rules or discontinuation criteria, both for individual subjects. So if there's a specific SAE, or obviously if someone becomes pregnant or anything else happens to an individual that they would need to come off of the study. You also need to really define the criteria for which the entire study would be stopped. So if there's a specific adverse event of interest, if there are a certain number of those within the population that are being studied, then the study would need to be put on hold, have the safety reviewed by maybe a data and safety monitoring board to determine whether or not the study could continue. So those types of things really need to be spelled out in the protocol. And again, I think I've mentioned this a couple of times, but if a subject discontinues the intervention, if they forget to take their medication, or if they decide they want to come off their medication, are they still going to be followed? Are you doing an intent to treat analysis? Do you want to follow all of the subjects? And at what rigor do you want to follow all of the subjects once they've stopped the treatment? Oftentimes, the protocols are written such that there might be a follow-up schedule for those patients, but it might be less rigorous than those that are still on medication. You want to have a section, and I know you guys have had a lot of mentoring by statisticians. So they're going to help you with this portion of a protocol, the statistical considerations. This is where you really describe your primary and secondary outcomes and how they relate to the choice of your outcome measures that will be measuring those. They're going to provide details on why a certain design feature was chosen. So if there's crossover or a washout period, the statistical considerations will list sort of the details of why. This will describe the randomization plan, sort of the criteria for stratification and how all of that was designed. And it will also describe the details of your sample size calculation. So again, your statisticians will really help with writing this section of a protocol. The next section is on data collection, site monitoring, and adverse event reporting. So these are the questions you need to think about, sort of how is your data going to be collected? If you're doing a single center study or a multi-center study, if you're doing a study that is going to be regulated by the FDA, you have to think about things like, does your system have to be Part 11 compliant? Does it have to have all of the bells and whistles so that if this study were a positive study, the FDA would look at the data and potentially approve a marketing application for the drug, or change the label of a drug. The other thing you have to think about is patient confidentiality. How are you going to blind the data so that it's HIPAA compliant, so subject confidentiality is maintained? How is your data going to be monitored? And there's multiple types of monitoring, and I think this is oftentimes a question. We monitor for quality, so it's typically a monitor that either goes on site or reviews data remotely. The data is also monitored by your project coordinator or your data manager, someone who's looking at the data to make sure that it's accurate. So it's the accuracy portion. Compliance, you want to make sure that there's been enough compliance. So again, all of that is being monitored by your by your study monitor. Also, how your safety is going to be monitored. Are you going to have an independent medical monitor reviewing serious adverse events in real time and adverse events on a weekly basis, monthly basis? Will there be a data and safety monitoring board? You need to list all of those details here in the protocol as well. You want to think about your subjects. The IRB is going to be reviewing your protocol. You have to remember that the IRB doesn't just do the initial review, but they also do continuing reviews. And what are the reporting requirements? And you want to list those in your protocol, just that you're going to be compliant with all of the regulations that are involved in IRB review and protection of human subjects. This is again where subject confidentiality comes in, making sure that you have your patients blinded, you have your patients confidentiality and you have a subject ID so that all of their identifiers are blinded to the investigators. You talk about if you're going to modify the medication or you're going to discontinue the study for any reason, how will you let the IRB know? You'll need to let the FDA know. You'll need to let your subjects know. You need to think about how you're going to be communicating with your subjects about that. Finally, sort of getting to the end of the protocol, and you want to talk about, how are you going to publish your research findings? You need to have a data sharing policy, or you need to have some input here about how you're going to share your data with other investigators. How are you going to share it with the scientific community? You should have some type of a manuscript plan. There are different manuscripts that come out of a study, methodology or protocol manuscripts. Those with baseline data only. There are ones for both the primary outcome as well as any secondary outcomes. You need to list all of your you need to state in the protocol that you're going to be listing the trial on clinicaltrials.gov, and you're also going to be compliant with having the results listed on clinicaltrials.gov. There are fines if you don't do that, so it's very important to list in your protocol that you're going to be compliant with that. And then, of course, you want to have your references of anything that you've cited within the protocol. It's sort of the last section of the protocol. So I'm now going to pass it back on over to Dixie, who's going to talk about sort of how writing this protocol. Oftentimes when you're writing the protocol, you don't have all the answers. You can't write all of it in a weekend. But by doing the writing part of the protocol, it helps to define where your holes are, and it can help you to better define your study logistics. So I'm going to have Dixie take on that.
Okay, thank you, Marianne. So Marianne's touched on some of these topics, but you know, once you're in the process of writing the protocol, you're really able to flesh out some of the very specific logistical considerations. And Marianne and I have a lot of experience doing this within NeuroNext and other projects that we worked on. And as she was saying, it's amazing the amount of things that come up, and it's almost like, you know, going through one roadblock after another. But it does help if you have some consideration ahead of time as to how to approach these things. So as far as drug supply, it's not the easiest thing in the world to come up with your drug supply. And there are a lot of manufacturers that when an investigator reaches out to them, they might be willing to help and provide a supply of drug. But you know, oftentimes the answer is no. Purchasing drug can be very, very expensive, and so that needs to be really determined before you go in with your grant application, because if you don't have a supply identified, your budget can just get blown out of the water if it's something that you're going to have to purchase, particularly if you need a matching placebo. You know, ideally those need to be coming directly from the manufacturer so that they are identical and matching. But there are studies where you can over-encapsulate. The downside there is there are subjects who participate who will open the over-encapsulation and try to figure out what that study drug is. But those are all considerations that need to come in when you're identifying your drug supply. You need to set up your vendor contracts and think about how much drug you're going to need. And expiration dates come into play here, because some drugs have very short expiration dates, and particularly if it's an IV preparation. Does it need to be stored at -70 or at you know, refrigeration or room temperature? -70 refrigeration requires, you know, something else to be considered, and that can be expensive, and it can be a space issue. So those considerations need to come in. And oftentimes, most institutions have a research or investigational drug service, but they might require fees to utilize those services. So you need to know what those are and consider what that is. Typically, I think it's a flat fee just to bring a study on board, and then it might be a per dispensing fee that comes into consideration. And of course, with the drug supply, you need to keep in mind what the adverse events are that might be associated with that particular drug. So for distribution, are you going to work with a central pharmacy, or is that going to be done locally? And this gets into the blinding aspects and the distribution and the labeling aspects. So if there might be supply that's purchased from a manufacturer, it might go to a central distribution. There need to be labeling to be compliant with the FDA regulations. And then also, if it's shipped out to states, there's across-state regulations that have to be followed. And then again, you know, is it able to be shipped or stored at what particular temperature, and what are the costs that go in there? There are some studies where the drug supply is limited, so therefore it's shipped on a per patient basis. Once someone has informed consent and screening, then the supply is shipped, and which requires a higher level of oversight to make sure that the site participating does have the drug available when they need it. And of course, following chain of custody for any investigational drug. And you know, the regulations get even more serious if you're dealing with a scheduled drug. The central laboratory considerations that need to come together. So, you know, we're all familiar with, you know, you order laboratories from your local lab, and the tubes go off, the results come back. But if you're going to use a central lab, which is often used in multi-center trials so that there can be continuity, because each lab has their own variations on how they report their results and what their coefficients are and those kinds of things. So you might use a central lab, which would require specimen kit assembly. And within the specimen kits, the tubes have certain expiration dates. Sometimes those can be short, so the kits will expire before they're used at the site. They might have barcodes affixed to the labels, which is actually a very nice way to do that, but that's an expense also, because then you need a scanner at the site and a reader. It's crucial if you're using a central lab and shipping specimens out to sites that the requisitions in the laboratory manuals are written very clearly, so that any participant on the study team is able to pick up those and process the laboratory, attain them and process the laboratories correctly and send them off to wherever the analysis labs are, and then also receive the results back. Other vendors that might come into play, central imaging and neurology studies, this is often a big piece. And MRI images can go a huge gamut from the type of scans that are obtained. So if the imaging is the primary endpoint in the study, there needs to be comparability. It can go from, we've done studies where there were actually dummy scans done on site, phantom scans, and then monthly QC scans that have to be sent in to make sure that the readings are coming across consistently from each site. Another thing that comes up when you're doing things like scans or laboratories is you might find incidental findings. So, perhaps you're doing a brain MRI for multiple sclerosis, looking for lesions, and uncover a tumor. How is that going to be reported? How is the IRB handling that? How will you report that to the subject? To make sure that those kinds of things don't fall through the cracks. And data collection. I have a big involvement with this piece, and there are, NINDS has what they call the common data elements, and there's a website about that which we haven't referenced on here, but if you put in NINDS common data elements on any Google search, you'll find their web pages. And they actually have data collection forms and variables standardized so that we're able to collect data consistently across studies in the same diseases. And so this is a great starting point for data collection if you're setting up a clinical trial. These things are all referenced within the data management plan, which should identify what type of data you're collecting, in what fashion you're going to collect it. A big recommendation is don't collect data that you're not going to use in analysis. It becomes a burden on the subjects. It becomes a burden on the sites who are trying to collect the data. It's attractive and people are interested in doing that, but it can really burn out a study and make it not as successful. Another tip is to limit open text boxes, because those are very difficult to analyze. So, drop downs, radio buttons, check boxes, those things all help collect data in a way that it's easier to analyze. So you also need to be thinking about your recruitment plans and your materials. And this is another place where the patient representative or patient advocacy groups can be extremely helpful. You need to think about this early, not later, because it can really help you, your project to be more successful. If you can partner with a patient advocacy group, that's extremely helpful. Actually ask them to read through a draft of your protocol and provide feedback. Also having them read through the informed consent form template is extremely useful. And the patient advocacy groups will often help you advertise and with recruitment, because they're always interested in participating and trying to move the science forward in their particular disease population. You do need to remember that you need IRB approval for any of the recruitment brochures or advertisements that you're going to use. Anything that would reach out to a subject needs to be approved by the IRB. And then, of course, there's the requirement for listing on clinicaltrials.gov. Another piece that comes in is pre-screening logs. As you need a partial HIPAA waiver to be looking through actual medical records to look for identifiable patients. Keeping pre-screening logs is a tool that you should describe to your IRB so that they understand what you're going to be doing. Because it's okay to keep some information so that you don't contact the same person twice, but you also don't keep that kind of information indefinitely. And it's also okay to structure a way to look for, what kinds of things are ruling people out from participation in your study, because it might lend to a relook at your inclusion-exclusion criteria, maybe there's too much rigor there or something that's going to preclude people from participating. So we would like to end with everyone should do a clinical trial, maybe more than once. You learn along the way every single time. There's the beginner slope, there's an intermediate slope, but there's the expert slope, and then there's a slope that ends up resulting in an orthopedic research study. But you can have a lot of fun doing this, and we hope that we've been able to give you some tips about writing a protocol so that you're able to operationalize all these pieces that come together. I think probably one of the biggest things Marianne talked about was that you're able to bring a team together to help you participate in that. So I believe we are going to take questions now. All right. We have a question here that says, do you need a HIPAA waiver for institutional-based medical repositories for pre-screening logs? I think anything that goes into a medical repository, yes, you're going to need a HIPAA waiver.
Yeah, you should check with your IRB, your local IRB, to confirm that as well.
Yeah. Typically, the waiver can be if you're just keeping it for the time of the study, and then you're going to destroy that data. But if you're keeping it in a repository, then that needs to be kept. And Will's adding that if you're looking at medical records, it's likely you need a waiver. Are there any other questions?
We have multiple attendees typing.
Someone's asking for access to the slides. I believe those get posted somewhere, but I'll let Sam follow up on that.
Yes.
Everybody able to read all these questions?
No, I don't think so. So we'll note that, yes, the slides will be posted.
So here's a question. NeuroNext is designed to help reduce some of the barriers to entry for clinical trials, in my basic understanding of it. How might the protocol preparation differ for NeuroNext trials versus those outside of NeuroNext or other predefined research coordinator centers?
Good question. I guess what I would say is, within NeuroNext, the coordinating centers, the clinical and data coordinating centers, we oftentimes provide standardized text for many of the sections of a protocol, because we already have standardized ways that all of our sites have been trained to manage different pieces of the protocol. We also have a central IRB within NeuroNext, so those sections are slightly different than in your typical protocol. So that's really, I think, the way most of it's the same basis of a protocol, and all the sections are still there.
And a lot of what we've described is for multi-center trials. There certainly are single-site investigator trials that don't require all of the sections that Marianne went through. We were trying to present this as, these are things that you should think about, whether it applies to your single-site study, whether you actually have a DSMB, whether you're, using a full-blown data system. Those things can vary quite a bit. And so you need to tailor it to the needs and the specifics of your particular project.
Yes, and I think that, as Will is now pointing out, having somebody that knows about databases and how data is collected, whether you're doing a single center study or a multi-center study, it's very important. You can't just save your data in an Excel spreadsheet and think about doing the analysis of that at the end. So you really do, you need to know how your data is going to be captured and really think about that, whether you're doing a single center study or a multi-center study.
And, you know, as far as like safety analysis and thinking about safety, there is, you know, even for single site studies, often like at my institution where something's within a department, all the players are involved. It's beneficial to even identify someone who's not working on the project, who's going to look at, you know, were there any adverse events that happened? So, you know, that can and should be done even on a small scale level, so that there's a little bit of an independent look at how the study's proceeding. You know, it doesn't get to the level of, you know, you're filing mid-watch forms and all those kinds of things, but it is a good plan for how you can set up your safety monitoring and make sure, you know, that that you're considering the protection of human subjects and also monitoring safety to look for any kind of signals that might be occurring.
Yeah, and one of the good resources that you need, if you don't have it within your individual department that you're working within, would be if you're working at an institution that has a CTSA, to work with your CTSA. Another question here that asks, for young investigators, what are some of the common mistakes that are made when designing trials? I guess my thing would be right up front is oftentimes investigators want to put too many things into their trial. So trying to bake a cake that has way too many ingredients in it makes the cake not rise. So I think that's a big piece. The other thing is I think investigators often very much underestimate the amount of time it's going to take, the budget that's going to be required, and they're not sure about how they're going to get their drug. And the other really important one that Robin Conwood is pointing out as well is that they oftentimes don't work with a statistician right up front. So working with a statistician to help really define what the goals of your study are, and making sure that you narrow down your questions. So again, you're not trying to do everything within your trial is really important. Yeah, some of the other things people are pointing out, too many outcome measures, too many variables, too many visits, too little time dedicated,
right?
Too little resources. I think when one of the things we work with investigators on quite a lot is putting together a budget, a realistic budget of how much it's going to cost to actually conduct the study. And another good one from Lori is not enough collaboration. Right,
and you know, this is another point where patient representatives can be helpful, especially on subject burden. Because, you know, when you're designing a study and thinking, you know, how great it would be to know these things and you know, this is an opportunity, but, you know, you pass that by a patient representative and they're like, you know, either, you know, if they're working or not working, those present different kinds of issues for people, but a full day of doing assessments at a site, and, you know, where they're coming from, distance, parking, all those kinds of things need to come into consideration when you're designing.
Yeah, I think Will just listed another one that I find myself running into a lot. Oftentimes, a junior investigator will think that their research assistant who doesn't have any experience in clinical research can also help them, and it's great because it gives them an opportunity to learn. But that means that they're expecting you, as the investigator, to be the experienced one and to really train them how to be a good project manager or project coordinator. So. If you're really new to this field, it's good to have someone like a project manager or a coordinator, protocol coordinator that's experienced, so you guys can teach each other things. It's really important to have an experienced project manager or project coordinator who sort of becomes your best friend and leads you through this process.
And that's another place where CTSAs are helpful. A lot of CTSAs are set up with a coordinator pool that can help. Money can be an issue again. You read it.
Yeah. So Will is telling us all that he was a coordinator for a trial at Michigan, and he only gave himself a C-. And I think that that's something that, you know, an investigator is an investigator, and they don't oftentimes have the same skills that are needed, or the detail orientation that is needed in the project coordinator role. So I think it's really important to recognize you need that other person who has that logistical eye.
There's a question: with extensive collaboration needed to help teach us how to run the trial, could we still be considered a PI of the trial? Certainly. I think that role is defined. I mean, you know, really, bottom line, the PI is responsible for the entire conduct of the study. We didn't go into some of the documents that state this, but like a 1572, you're actually signing there that you take on full responsibility, which is, you know, why, you know, because of all this, we're trying to point out that you need all these things in place to make it successful, because no one person can do all those things. You need the collaborations, and you need good people to help you do those things. But the PI does retain ultimate responsibility for the study.
And you're the one that thought of the question. You're the one that came up with the hypothesis. You know, so it really is your study. It's just you need people to help you to operationalize it. You need the statistician. You need all of these different team members to really make your dream a success.
And Will's also pointed out another point here about retraining, and this is something, you know, from the coordinating center perspective that we are constantly involved in. There is turnover. You know, if you can hang on to your team the entire time, that's awesome. But you know, and you know, if you're involved multi-center, there's often retraining programs. You know, things like this course and being able to post these videos can help with that, or specific to the particular project as well.
So I hope everyone can hear me. Thank you, Dixie and Marianne, for a great talk. And thank you, everyone, for joining us for this webinar. I just wanted to point out again the 3 links at the bottom of your screen. The first is to the course website, so you can find more details about the course, and previous webinars are posted there as well if you are interested. The second link is our webinar evaluation form, so please make sure you click on that and give us feedback about this webinar. And the last link is the protocol template link, which was discussed earlier on in the webinar. So thank you, everyone, and the next webinar is in the advanced track, and that'll be on June 24th at 1 p.m. Eastern Standard Time. And the next foundations track will be on the 26th at noon Eastern. So thanks again.
Thanks, everyone.
Thank you.